So we have with us Mr. Todoro as a part of the scheme of the GIAN program, where our research scholar, faculty, and the students they get the opportunity to interact with any foreign faculty in certain domain areas. And I'm happy today the man with us. I think he is in the area of probiotics. Very interesting area. Uh, we normally deliver knowledge to classroom lectures. I'm happy to know that out of 12 sessions which has been designed by Professor Santosh Tiwari for the students, research scholars, and of course the faculty, 8 out of 12 would be lab-based kind of a interactive session or a hands-on training kind of things. Lecturing is important, but more important is questioning from the side of the students and research scholars. I think Professor Toto would be more happy if more and more questions come from your side after listening to him. And I know our students, especially the science students, they are not only active, agile, and intelligent crowd. At the same time, they know how to best utilize their time. Don't be a passive listener. From my side, I would advise you don't be a passive listener only, be an active participant for this workshop. We at MDU, we have a lot of emphasis on the capacity building of our students and research scholars. And this GIAN program is one of the approach of the university that how we can take a benefit of various schemes launched by the government of India for the exposure of for the international exposure of our students. You all know that we are the only university where 1 week workshop at the time of the moment a research scholar is enrolled with us as a research scholar, we try to initiate the research scholars in research methodology, and 1 week workshop, which is rare, and I think we are the only university we started some 6 years back. It was the year 2019 when I was given the charge of this university in addition to the charge of the Vice Chancellor of SOPOA. We started that workshop, and the research workshop were from A to Z kind of a thing for research or hands-on training in research areas, and that program has become very popular. And of course, that has improved the quality of our research scholars. The same way, I know that prior to this workshop, we have organized in past 2-3 years so many GIAN programs. Santosh Tiwari is the coordinator for this, the nodal teacher for organizing the GIAN program. I'm happy our life sciences department, they are doing good. And there is no denying the fact that our life sciences department must be among the top 10, if we talk about top 10 universities in life sciences, and our pharmacy department, they are the best department, they are the flagship department of this university. And I take pride in this fact that our students are doing very good in this area, in the life sciences area and the pharmacy areas. The teachers, the research scholars, all are hardworking, and I compliment the efforts of teachers in bringing our life sciences department and pharmaceutical sciences department to this level. And on behalf of you all and on behalf of the university, I welcome Professor Todoro, and I'm confident that your interaction with this international faculty will expose you to international standard or international approach to various the problems, more specifically the probiotic issue as explained by Professor Santosh Tiwari. We have planned so many other international exposure to our students shortly in the month of November and or maybe in the beginning of December. We are organizing a big program of MDU Global Connect, where our students who passed out from this university, our alumni department, they are organizing a program, MDU Global Connect, where our students who passed out from this university and they are doing good in different areas, be it medical or be it life sciences or any other area of the knowledge, they will be invited in the month of November or December, and they would be interacting with if somebody belong to say environment science department or somebody from pharmacy department, they would be interacting with the students of their respective department where they study, and they have offered, some of them even in advance, they have offered hand holding of our students, the mentoring of our students. This will give opportunity to students connect with the international faculty. So this is another or the next level of the university where we are trying to connect our students with the foreign faculty for their international exposure. So with these words, I compliment Professor Santosh Tiwari and the entire team of the genetics department led by the Dean Faculty, Professor Meenakshi, and of course our Dean Research and Development, Professor Teresa, and Director IPSC, and the entire team led by our Dean Academic Affairs, Professor S.C. Malik. So my compliment to the team MDU for organizing this wonderful GIAN program. And I'm sure that it's going to help you in a long way and exposing you to the international faculty. Thank you. Thanks a lot.
A sincere thank you, sir, for your insights, thoughtful words, and for gracing this event with your presence and wisdom. It is now to honor our esteemed guests with mementos and shawls in the recognition of their gracious presence and contributions. May I request our Honorable Vice Chancellor, Professor Rajveer Singh, to do the honor of presenting a memento and shawl to our guest, Professor S.D. Todoro. I would now request Professor S.C. Malik, Dean Academic Affairs, to kindly present the memento and shawl as a token of appreciation to our Vice Chancellor, Professor Rajveer Singh.
ठीक है समझ मन यार क्या करूं इसको समझ कहने हो रही छोड़ के किट गए अरे बैठ जा भाई बुरा दिया सॉरी भाई डर गया सॉरी छोड़ दो भाई छुट्टी हो गई टीचर को बोले बरासी
Vice Chancellor has few urgent assignments, so need to leave. I request Professor Harish Pureja, Dean Research and Development, to welcome our Dean Faculty of Life Science, Professor Meenakshi Vasisth. Dean Faculty of Law. Now, I with these words, I invite Professor Meenakshi Vasisth to say a few words about this program. Welcome.
A very good morning to all of you. And on the behalf of Faculty of Life Sciences, it is my immense pleasure to welcome you at the inaugural ceremony of one-week workshop on lectureship of probiotics. As our Vice Chancellor has left due to some urgent requirement, anyhow, I express my heartfelt gratitude to our worthy Vice Chancellor, Dr. Rajveer Singh, who had spared time from his busy schedule and graced the occasion. I also welcome our Dean Academic Affairs, Dr. S.C. Malik sir, Dean Research and Development, Dr. Harish, Coordinator of GIAN Program, Dr. Narsimhan, and I extend a warm welcome to our distinguished speaker, Dr. S. Todorov, to our university. Thank you, sir, for accepting our invitation and making this event possible. And we are fortunate enough to conduct this GIAN program by accepting our invitation. Dr. Todorov had made it possible. And briefly, I want to apprise about our Faculty of Life Science. We are 10 departments with more than 50 teaching faculty and more than 250 research scholars as our research team. And they are doing excellent in the covering almost all the areas of life sciences. And we are running graduate course, postgraduate course, diploma course, certificate course to engage our students. And we have distinguished publication in various national and international journals of repute. And I am proud to share that our scientists have made us proud. I want to mention the name of Dr. Giri, Dr. K.K. Sharma, Dr. J.P. Yadav sir, Dr. Jahan. They are among the 2% scientists of the world and making our university proud. Now I want to congratulate Dr. Narsimhan and Dr. S.K. Tiwari and his entire team for organizing this workshop on a very important area. Everyone is struggling to deal with the MDU strains of pathogens, and there is continuous increase of use of antibiotics and pesticides. to combat the infection and leading to disease resistance teams and posing a threat to the human sustainability. The present workshop is a golden opportunity for our young minds to explore the horizons of bacteriocins and to develop protein-based natural antibiotics for the human health. It is the need of the hours too. Now, I want to say that, dear participant, enrich yourself, make the most of the workshop, and contribute towards a better society. Make the earth a better place to live in for yourself and for your future generation. Wish you all the best and a fruitful interaction with a fruitful outcome. Thank you all.
Thank you very much, ma'am, for your insightful and inspiring address. I would now like to invite Professor Harish Durija, Dean R&D, to kindly address the gathering. Welcome, sir.
Good morning to one and all present here. Professor S.C. Malik, our Dean Academic Affairs, Professor S.D. Pradao, the foreign faculty for this GIAN program, Professor Meenakshi Vasisht, Dean of Faculty of Life Sciences, Professor B. Narasimhan, Director IPAC, the GIAN coordinator of this program, Professor Santosh Tiwari, faculty members and dear students. This university was established in 1976. In the last 50 years, a major impact has been made towards research, which encompasses mainly, as Vice Chancellor has rightly pointed out, the Faculty of Life Sciences. So the Faculty of Life Sciences and Pharmaceutical Sciences are doing good research work into this university. Along with all other departments, we have such a research ecosystem that we have different policies in place. We have R&D cell, which was established as per the UGC direction 3 years back. And we have the research promotion policy, IPR policy, consultancy policy, as well as innovation and entrepreneurship policy as well. So if I look into the research ecosystem, we have the IABET Center Instrumentation Laboratories. We have encompasses a number of instruments, sophisticated instruments for the research work as well. In addition, we have the separate animal house for the animal experimentation part, as well as human ethics committee for if you are looking for the work on the human participants. In the last 5 years, the projects are worth more than 25 crores, more than 8,000 publications in the different Scopus and Web of Science journals, including the 1 crore worth of the consultancy projects, as well, and filing 75 patents on the behalf of the university. So this university is doing towards this research an excellent opportunity for the research scholar to learn and go ahead. And I can say the GIAN program is one part of the same, where we have the more opportunity to interact with the foreign faculty, having the more insight what is going outside India towards the research. I hope this 1 week program will give you more insight on this program as well as the skill training into the lab. Thank you so much.
Thank you very much, sir, for sharing your valuable words and insights with us today. It is my distinct privilege to invite Professor S.D. Todorov to the dais to share his insights with us. Please, sir.
Good morning, everyone. Dear respectable authorities from the university, dear students, it's a very big pleasure for me to be here. It's not just a pleasure, it's a privilege. Because something that I believe that the science needs to be open, thinking needs to be for everyone. And in fact, students, you're the future of the science because you're the next people that will discover the new things, new events. In this workshop, actually, when we built the program with Professor Tiwari, we was planning large options, but we pay a very special attention on interaction, on the practical work, because in fact the workshop is meant not just transmitting knowledge from one to another side. Actually, we will be transmitting knowledge from the both sides. Do you think I'm not going to learn from you on this practical work? Of course I will. And well, I will try to show you what I know, what is my experience, because with the time of working the lab, you will experience. But again, a lot of ideas, a lot of new points that could be explored actually coming from you to me. So it will be two-directional interaction, and I hope this will be a good option for you to see what I win in my experience and what I actually can learn from you. So thank you very much for the very hard welcome here, and it's really a pleasure to be in my first trip to India. I hope not is the last. And it's, again, maybe I will convince some of you to come to work in my lab in Brazil, because I'm Bulgarian, I'm not Brazilian, but my heart is in Brazil. And it's a nice place to explore the country, explore the science. University of Sao Paulo is considered one of the biggest in the Latin America, with almost 200,000 students. Campuses all around the state of Sao Paulo, because it's a state university, it's not a federal. But it's one of the home of the largest institution investing in the science and education, because I mean, education is more important. This is the basic of everything that can be done in the future. And the good science actually starting in the first year, and even in the school and then in the university, and is building all the time on the top and the top, and giving a pleasure when you discover something, and giving the pleasure to know, yes, I have done this. It's my work. My work finished to this patent. My work finished to this medicament. My work finished to this process. It's improving the life of everyone. And the pleasure of the discovery is a process. Thank you very much for the welcoming, and hope to enjoy. You will enjoy the lectures and the practical works in the next week. Thank you.
Thank you very much, sir, for your thoughtful words and for gracing this event with your presence. Now, I heartily welcome Professor S.C. Malik, Dean Academic Affairs, to share his valuable thoughts with us. Welcome, sir.
A very good morning to all of you. Respected Professor S.D. Todro, Madam Meenakshi Vasisht, Professor Harish Reza, Professor Narasimhan, Professor Santosh Tiwari, and all other professors of the Department of Genetics and Life Sciences. Really, it is a great opportunity for the university to organize such type of wonderful workshops in the interest of the students. You might be knowing that such type of programs were initiated in the year 2015 with the objective to connect or to interact with the foreign speakers or scientists, so that we can be in a position to understand what they are going to do. Since 19, as our Vice Chancellor Rajvi Singh already stated, that we are conducting such type of program at our university level. This is one of the wonderful universities in India and very close to Delhi. So far as science subjects and science departments are concerned, our science departments are dominating in both research and teaching. I have been observing this last many years. Because of these persons, we are far ahead, and we are at the top in Indian universities. And we have somehow the status or the status in world universities also, because of the birth of these persons from the life sciences, pharmaceutical sciences, and other sciences. The purpose of such type of program is to connect ourselves, global interaction, global initiative of academic connections, networks. This is GIAN. Because we are here only to listen our teachers, our research scholars, our scientists, but we are not much aware what is going in the world. So such type of programs were initiated because of to know, to interact with the persons who are working throughout the world. So here Professor Todro is sitting with us because of the scheme of the government of India, particularly the Ministry of Education. We have several departments who are conducting such type of programs. Dear students, it is a good opportunity for you to interact with the persons like Todoro. You have the chance to interact with him to clear your ideas about the research. You have the chance to interact with him to know to improve your skill, how to teach the students in a good way. That is the good methodology. Our university is trying to give best solution for such type of programs. Since last 7 years, I have been observing that our university is organizing many programs in the interest of the students. Maybe into the research methodology workshops, and maybe some training programs, maybe hands-on training programs. And during these 6 days, you are going to interact with Todoro through, I think, 12 lectures are going to be delivered. And in all these lectures, you will have some hands-on training programs. I hope such type of programs must be the part of your curriculum also. We know that you are good researchers, you are the budding researchers of our country, particularly our university. So this is entirely the program being sponsored by the Ministry of Education. And Professor Narasimhan, Professor Dureza, and many other professors are trying to give you the best solution so that we can improve our skill, improve our knowledge, improve our teaching methodology, so that we can excel throughout the world. The purpose is to make our students so that they can represent our university, our country throughout the world. This is the chance. I hope that during these coming 6 days, you will enjoy the lectures, the interaction with Professor Todoro and all other speakers. I don't know who are also coming here, but this is not the program only to interact with the international scholars, but also we can have the chance to interact with the scholars, the renowned scholars from national level also. But this time I hope Professor Todoro has been requested to give something in this particular area. So on behalf of the university, I congratulate in advance you all. I'm very thankful to you, Professor Santosh Tiwari, and all other teachers of the Department of Genetics and Life Sciences for inviting me to interact with my students, coming researchers. Thank you all of you. Best wishes. See you once again. Thank you very much.
Thank you very much, sir, for such encouraging and meaningful words. Now, I would like to invite Professor Santosh Kumar Tiwari, Head Department of Genetics, as the co-coordinator of GIAN program for the Board of Science.
Thank you, Divya. I think it's just a formality before the contribution our dignity has given. Vice Chancellor has no time, but he gave some for us. He came, Professor Rajesh Singh, Vice Chancellor of this university. Thank you very much, sir, for sparing time with us. Professor S.C. Malik, Dean Academic Affairs, he was busy with the meeting, but when I called, we managed it in a very short time. Sir, I'm very thankful, and your words are really inspiring to the participants. For next 1 week, they are going to attend it. So we need this kind of inspiration to the students. I'm very thankful to Professor Narasimhan P. He was actually a genesis of this program we started, and keep on emailing that applying for this GIAN program. I was neutral, okay, I will do, but keep on consistently that you should before the deadline. So because of his consistent effort, it was possible and manage all those technical difficulties during the application. Thank you very much, sir, for the support. I'm thankful to Professor Harish Dureja, Dean R&D, and and he was always with the motive that we should inspire and motivate our research scholars to do better. And thank you very much, sir. Professor Meenakshi Vasist, ma'am, she is our senior of the department and also at the brilliant Dean Faculty of Life Science. I am very thankful for her motivation and program organizations, all the inputs of the organizing even small steps to this program. Um, there is no words to well thanks Professor S.D. Tororo because his efforts to come physically is something inspiring for our university and our students as well. You'll be surprised to know it took 36 hours continuous flight from Brazil to India from Paris. So he was tired, but looking at the importance of this program, he thought that no, I will be there at the inaugural session and come. So thank you very much, sir. You accepted our invitation and took this effort because for a short time duration it is easy to fly, but 36 hours, even I fear to go or not. Sir, it's a really great pleasure for our university. And all the university officials have heartily welcomed him and provided all the hospitality to the university. Thank you very much, sir. The coordinators from department, Professor Ritu Yadav and Professor Neelam Sehrawat, Dr. Mukesh Tanwar, this is a teamwork. They always are coming together, interacting that this program should be like this. Always, because although there was a vacation, so due to vacation even they were in touch how to plan it with even small minors, all this. I'm thankful to all the heads of department taking, for the schedule they have left now, but I'm very thankful to those head department of pharmaceutical science, head department of microbiology, food technology, botany, zoology, biotechnology, microbiology, all the visited and came, all the faculty members, Professor Rajiv, Professor Rajiv, and all these faculty members, they came and inspired with us. From the AIMS faculty, they came Dr. Ritu Agarwal with us. She was interested to join the program. Professor Yadav is with us. So all they spared their time. And in the last, the students, they keep their interest to participate in this program. So thank you very much to all of you, one and all, even at least the name of everyone. Thank you very much.
Thank you very much, sir, for delivering such a warm and meaningful vote of thanks. And now it's a moment of pride and unity as we pay homage to our nation. I kindly request you all to please stand on your seats for the national anthem.
तव शुभ नामे जागे, तव शुभ आशीष मांगे, गाहे तव जय गाथा। जगन मंगलदायक जय हे भारत भाग्य विधाता, जय हे जय हे जय हे जय 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 हे।
Thank you all of you for your attention. We will now take a short break. So I invite all the dignitaries and all present here to join us for a delightful high tea.
here, or few of you are still outside? All right. So, did you message from the WhatsApp group? We will start in 5 minutes.
Okay.
Okay. So, all right. So, here we go. The first talk of this workshop, the innocence of probiotic bacteria, from food safety to human and veterinary health promotions. So, I hand over the stage to Dr. Dodro to continue. Thank you so much.
Okay, let's give few minutes to other come. Before the people actually coming, please, I want to know about you. I know that there is interaction. It's not just me talking. Everyone is from this state? Someone from another state? No one? Okay, Haryana is supposed to be one of the best ones in this condition. They already told me that this state is giving most of the Olympic champions in the India. Okay, so, so do you want me to start already or wait a little bit?
You can have a brief introduction of each of the participants.
Okay, maybe this is a good idea.
Start from here.
Okay, we're starting from you. So you're the first line. Just give your name, what is your area, your undergraduate, master, PhD. If you are in like PhD or master, just a very 1, 2, 3 words on your topics of interest.
So my name is Sarika. I have completed my master's in human genetics. Currently, I am pursuing PG diploma in genetic counseling. That's it.
Thank you.
I'm Jagvir Rawal. I've been head of immunology at Lala Rajpatra University of Veterinary Sciences at Hisar in this state. And presently, I'm heading our innovation research and development cooperative at the national level. I was interested in this veterinary and human health, One Health.
Well, we need to think about the medicines for the human. And if we keep the animals healthy, it's even better. It's better for us. Okay, let's go for the next one. Oh yeah, they arrived too. New arrival. Okay, but maybe then you go first, and then the girls will present.
My name is Dipanshu. I am currently pursuing my BSc Genetics.
My name is Bhavna. I am currently pursuing BSc Genetics.
Be louder, louder.
I am Dharmi. I am currently pursuing my BSc in Genetics. Myself, Kirtika. I am proceeding BSc in Genetics.
I am Nidhi, a research scholar from the Food Technology Department, and currently I am doing work on basically celiac disease and the products we can prepare using the hydrocolloids and gums.
I'm Umang Solanki and I'm doing my PhD in food technology, and my current topic is based on cellulose, which can be used for the alternative of the plastic packaging films.
Hello, myself Parul. I am a research scholar in the Department of Forensic Science. Currently, I am working on the seasonal analysis of entomofauna and microfauna associated with buried pig carcasses.
I am Muskan. I have completed my graduation from Ramjas College, University of Delhi in BSc Honours Zoology, and currently I am pursuing Masters in Genetics from Department of Genetics, MDU.
So we're going to give a very difficult time here. Everyone in genetics, and it's a difficult area.
I'm Himanshi. I have already completed my graduation from Kishore College, Juniper, which is affiliated to MDU. Now I'm currently pursuing my master's from genetics of Department of Genetics, MDU.
Good morning, sir. My name is Monica Yadav. Currently, I am pursuing my PhD in microbiology, and my research topic is generation of bioactive peptides by enzymatic hydrolysis of plant-based proteins.
My name is Kritika and I am pursuing PhD from Department of Microbiology, and my research topic is to explore the bioactive potential of therapeutic potential of bioactive compounds through purification.
Great.
Good morning, sir. My name is Arush, and currently I am a research scholar in the Department of Microbiology. I work on the isolation and characterization of probiotics for treating inflammatory bowel disease.
Good morning, sir. is Samriddhi and I am pursuing my PhD from the Department of from Center for Medical Biotechnology. And my work focuses on screening probiotics for therapeutics in celiac disease.
Are you taking probiotics yourself?
Yeah, I do, in form of natural sources.
So you're the first one that tried them to see if they work.
Sir, my name is Keerti. I have completed my master's in microbial biotechnology from Department of Microbiology, and now I'm doing training in regulatory
Can we do later? Can you do later? Because, you know, we don't have time.
Okay. Okay, we can continue in the next.
Rest we will start from there only. Welcome back to all of you. Sit in the front. I think all these rows are freely available for you. You don't have to pay. So
who is asking more questions with the chocolates?
Yes. In between you can. So we are going to start this technical session. Really a kind of inspiring, motivational, and historious connection with Professor S.D. Todorov will take you to the field through a different story. So you will be surprised whether I'm studying or I'm listening a story. So it's a very interesting. The way of his teaching I like to the students, they really connect it. So with all this, I invite Professor S.D. Todorov to deliver his talk and technical session. There will be two lectures from here. One after one, there is short break, and then we can do it, or if you want two lectures continuously, depending on your idea or your solution. If you want, you can continue for two lectures and then lunch. After the lunch, there will be a practical session, as an interactive session in Department of Genetics seminar hall. You can come after the lunch directly. This session will be chaired by Professor Rajiv Kaur. I invite Professor Rajiv to have on the dais. If you want to listen the lecture, you are comfortable, you can sit it. Professor Rawat, come on the front side. You can sit with the sofa. Professor Rawat is from the veterinary science and interested to listen this course. And thank you very much. The dais is Professor Todorov to inspire. Welcome, sir.
Okay, let's start. Because if you want to finish, we need to start, no choice. And if I don't finish, Santosh will punish me. Okay, normally I say to the people, someone that is able to pronounce my name and to read my name without ever having won higher marks in the exam. No one till now managed to have these benefits because the name is quite difficult, Svetoslav. But I mean, it's typical Bulgarian. It's Bulgarian, it's normal, but the outside is different. Okay, where I'm coming from? I'm Bulgarian. I'm born in a small town in Bulgaria, in northwest of Bulgaria, in 1970. Then I had studied at Sofia University. In Bulgaria, normally when you start studying, you're studying microbiology from the first year. So it's like 5 years microbiology, no choice. And we need to have the biochemistry, we need to have the genetics, we need to have the mathematics, physics, chemistry, etc. So end of the day, I finished in France, where I made it my master, and then my PhD in the exchange program sponsored by French government. Well, then finish my PhD, what to do now, where I can work. When I'm born, I was born in the communist time, then I started studying already was democracy. I don't know if it's the right name to call it, but they call it democracy. But there was no jobs. And I started working in a private company as a research director of a winery. It's the only one winery in the world outside of France that's allowed to produce champagne. And in the beginning, I was thinking I can drink all the champagne as a winery. No way, you cannot do it. And you don't want to see champagne after the second week. Well, some people think it's exciting to do nothing, just drink champagne every day, and to do nothing really in the company. The reason, at that time, Bulgaria was not part of the European Union. So we needed actually to have microbiologists as a full-time job in the company in order to export the wine. You see, it was a good profit. I had a car from the company. I had nothing to do. good salary, but I was feeling boring. So I decided to go to South Africa when I went as a postdoc. In the beginning, after that, I became a researcher in the Stellenbosch University. And after Stellenbosch University, moved to Brazil, was invited there for a short time, but then I stayed like almost 20 years now. Then I was working initially in the Sao Paulo University, then in Viçosa University, go back to Sao Paulo, then come the opportunity to go to South Korea, and I moved to Pohang University in South Korea with the idea to stay 1 year, but then I stayed 4 years because of some pandemics in the between. And then here in the bottom, I don't know if you can see at the bottom part of the slide, but you can trust me. I passed some concourse and I become a full professor at the University of Sao Paulo. So now, no choice, I need to work in Sao Paulo because it's my... In Brazil, we have a joke. You need to explode the university to kick you out of the university. But if no one saw this, you can still work in there. So, but anyway, we need to produce, no choice. They're asking the paper, they're asking the projects. So, part of the... Okay, you can call me by my short name, Slavi. It's much more easy. In Brazil, again, people call the professor on the first name. Here in India, it's in the last name, but Latin American system, it's using the first name. You have my 2 emails, the private one that I'm using for most of the publications, etc., and my university email. So if you have questions, you can feel free to send me. On the first lecture, in fact, now we're going to finish with the fun, we're starting with the work. We're going to talk about the lactic acid bacteria, their taxonomy, biochemistry, identification, and then we're going to bacteriocins, etc. So following the program. What is lactic acid bacteria? Who worked with the lactic acid bacteria? Hands. Yeah, some days good. So make my life a little bit easy. Okay, can you hear me if I talk like that without microphone? Or you need a microphone for the record? Do you need a microphone for the record or not? Okay, so sometimes, okay, but I will try to keep it in the mic. Maybe it's better. Well, if you think about lactic acid bacteria, they are not systematic group. This is clear. This is not a genus, this is not a species. This is not systematic group that can have some rod, that can have some copy. And inside the rods, you can have it lactobacillus, that actually we cannot call anymore lactobacillus because they changed their names. Remember, in 2020, they reclassified them in the several, I think, 23 different genes. Well, this makes life difficult. You already tried this new systematic lactobacillus, lactic acid bacillus, bombibacillus. Well, not a bomb. Bombi is because of Bomba. It's a scientist from Slovakia. We have a whole subphalia. Most probably who worked with lactic acid bacteria already saw the name Wilhelm Hotzapfel. He's one of the biggest researchers on our time working with the lactic acid bacteria and really involved in the taxonomy. But as well, in this group of the rods, we have the different carnobacteria. In the cocci, the life is much more complex because they will have Lactococcus, Enterococcus, Pediococcus, Leuconostoc, Vicella, Pediococcus. And it's interesting, Streptococcus, yeah, it's there. Interestingly, work with the lactic acid bacteria, it's a very nice because you have the option to discover the new species. If you work with the botanics, I will have chance to discover the new plant. Maybe it's a chance. With the animal science, the new animal species, well, the people was already described almost everything. Well, recently there was a new snake discovered in Amazon. Luckily, not poisoning, because I have phobia of snakes. Honest with this. If you work with the lactic acid bacteria, because the taxonomy was based mostly on the physiology and the biochemistry in the past. But then coming the genetic methods inside, you have the much more precise way, and something that is believed is the same genus or same species. In fact, you realize they're different because from genetical research you can see that it's different. So these days you have this Mencia, Isobacillus, Paralactobacillus, Tetragenococcus, Oinococcus, Melissococcus, etc. Even sometimes in the one genus you have only 1 or 2 species. This is the story with Oinococcus. They separated Oinococcus from Leuconostoc because of genetical differences. And after it's only one Oinococcus only that is part of this genus. But now it's coming the point. If you want in the microbiology, you can discover the new species. And then you can explore these species and you can see if the species that you discover, this strain that you discover, is actually safe or virulent. And if it's safe, you have, well, anyway, you have right to give the name of this new species. So if it's safe, you can give it your name if you want, no problem at all. You can make your name in the history, but if it's a dangerous, pathogenic species, you can give it the name of your professor. So you're giving the revenge to them for difficult time in the lab. Okay, in Brazil, normally I say you can give the name of mother-in-law, because normally mother-in-law in Brazil is no one like it, not in Bulgaria. It's India, you like a mother-in-law? I don't know. You have right to like it, but keep in mind you have pathogen species discovered. You can name it the kind. Well, what the lactic acid bacteria are? We still continue this question. Okay, they are gram-positive. It's for sure they cannot be gram-negative. And I'm getting very upset when I see in the literature that lactic acid bacteria are gram-positive, gram-negative. No way. They are only gram-positive. They can be immotile, acid tolerant, they're also okay spheric organisms, and all of them are catalase negative. Why this is important? Because when they're catalase negative, they need to develop the new mechanism, different mechanism to cope with the oxidative stress. And they're producing antioxidants. And these antioxidants later we can actually explore like a beneficial property of this lactic acid bacteria. Because the difference, if you take bacillus, like Bacillus subtilis or Bacillus belezensis or whatever bacillus, they producing catalase. So very often find the bacillus that producing antioxidants is not such an easy way because they have different, more cheaper, more easy mechanism actually to reduce the oxidative stress. But lactic acid bacteria, they don't have options. They cannot produce catalase, so they need to have antioxidant properties molecules that they will use to protect them. Okay, and this is coming actually very figurative examples of this, how the lactic acid bacteria are. Actually, you see, here we have the bacillus. They are not part of the lactic acid bacteria because lactic acid bacteria belong to the order Lactobacilli only. They cannot be confused by bifidobacteria because they are part of actinobacteria. And they are not lactic acid bacteria. So please, when you write your papers, be very careful. Don't say lactic acid bacteria are lactobacilli and bifidobacteria. No. Bifidobacteria don't belong to the group of lactobacilli because they are not part of this order. They are part of the class Actinobacteria. Firmicutes are part, lactic acid bacteria part of Firmicutes, but order of Bacilli are not part of the lactic acid bacteria because they are catalase positive. So lactic acid bacteria can be rods, can be cocky, can be, they don't produce spores. Then actually it's a good or bad. We can discuss this later, spore production. But they are not lactic acid bacteria, no spores, they are not catalase active. Okay, and then coming this program in 2020, the 23 new genera being described from the former genera Lactobacillus. The reason. Again, how many people was mentioning that they work in genetics? Genetics mess with the taxonomy of lactic acid bacteria because they just simply show that it's enough genetical differences to be considered all these new genuses as a part of the same genus. But now is coming a problem. You write a paper and you want to refer to something that is before 2020. And you want to refer to Lactobacillus because before 2020, Lactobacillus was one genus. Now we have 23 inside. How are we going to refer to it? You just simply use the English word. You don't say Lactobacillus, that is Latin, that needs to be in italic because it's not the English, and need to be with capital L. So you just put simply English word Lactobacilli, not italics, not capital letter. And this is actually recommendations from the authors of the paper that they was actually looking and predicting that this problem will arise. So the correct way. But then is coming another problem. How I'm going to abbreviate in the paper because Okay, I presume everyone already published a paper. No choice. If you want to go the progress in the academy, you need to publish. And you need to publish more and more and more. And even some programs, they request to have the PhD, you need to have at least 2-3 papers already published. Sometimes it's making a lot of pressure because the coming nasty third reviewer saying, I don't like the paper, you can make another experiment. Well, I made it animals experiments, I don't have any more animals, how I can check something else? Well, but the reviewer insists. Okay, before was easy, lactobacillus, okay, LB or L, fine. Now we're having a huge variety of names. So in 23, with some colleagues, we proposed a standardized abbreviation. So for each of these new 23 species, genera, we suggested a different abbreviation in order, because when you you read the paper, you see L monocytogenes, L innocua, L lactococcus, L plantar. Well, but this L is not standing for the same thing. Could be leuconostoc from Ascaris, could be Listeria, could be Legionella, could be Lactococcus, Lactobacillus, Lactiplantibacillus, Lacticacibacillus, etc. So abbreviation, and very often the technical part of the journal is giving you a hard time because you submitted everything, you think everything is going well, and then you need to correct again and again and again, and your supervisor is getting on the top of you saying, why you don't make it in the beginning? Well, I'm a student, I'm still learning, you're the supervisor, why you don't tell me? But again, you know, it's not only teaching in the supervisor, we need to write a project, we need to apply for money, we need to go to India, we need to go to Taj Mahal. Well, I went there on Saturday. So you have this kind of recommendation. And then these are some examples. I don't know if the contrast is, maybe can we try to stop these lights here in the front, or it's not possible? All the lights in the same system. If it's possible, please, I will appreciate it. Maybe we'll be the better contrast. So we have these all the names, and we have the standard abbreviation. We suggested like 3 letters, we suggested 2 letters. There is an option, it's easy, paper is available on the internet, it's for free. You don't need to pay for this. And in this way, you can download and you can make your life a little bit easier. But now, as I mentioned, the lactic acid bacteria, they're producing acid. Well, other bacteria are producing acids as well. Not really. Even Listeria producing some acids as a final metabolite, and it's not lactic acid bacteria. And especially people working in the food safety, they know how the nasty could be Listeria monocytogenes. But important thing, in lactic acid fermentation, process of the transforming of glucose, first we're going to have a pyruvate. If you have this process and then pyruvate going to lactate as a final electron receptor, this is a typical pathway in the lactic acid bacteria. There is no way to give it in a different way. And then coming the problem, okay, lactic acid is a final metabolite, but then nature can get a little bit nasty sometimes because they can produce 2 forms of the lactic acid, N and D lactic acid. Well, very often people checking just the pH and saying, well, we have the good lactic acid ratio. Well, it's not true because pH is one story, production of acids is a different story, and we don't have choice. We need to go chromatography or need to have a kit for the detection of the lactic acid specifically because we need to know the level of the lactic acid production. And especially we need to know if it's producing only L or D form. The problem is when you have too much of D, our organism is liking L forms, like L amino acids, L lactic acid. L lactic acid can be metabolized much more easily compared with the D lactic acid. And especially for the babies and for the kids, microorganisms, probiotics, they are good, but they're not good for everyone. Because some probiotics that producing maybe a little bit more of their lactic acid could be very problematic for a baby. For the human, like an adult, it's not a problem. But for the babies, could be, it's better like that? Yeah. So now we can sleep, but just please don't snore. For the camera, it's okay, the light? Because we need to respect the rules to be record. There was Lactobacillus rhamnosus, very good probiotic. There's a Lactobacillus reuteri. Reuteri is on the market as a probiotic, Lactobacillus reuteri. So its new name is Lactobacillus reuteri. You see, even I'm struggling with the new names, but well, need to learn them, of course. Lactobacillus reuteri is commercialized as a probiotic, but this strain, by its nature, is producing a combination of L and D lactic acid. Even if you open the Burgi is like a Bible for the microbiology because everything is well described and systematically explained. They say they're producing almost equal quantity of L and D form of lactic acid. So you're obviously not supposed to give this probiotic for babies. And do you know what? I found on the market drops for babies containing Lactobacillus reuteri. I mean, these people, they developed this product, they never opened the book of microbiology. What's happened? Because industry, sometimes they ignore it. They want to make a profit, and they ignoring basic rules of safety. Sometimes I like, actually not some, always I like safety is priority. When we work with the lactic acid bacteria in the lab, first step is to check the safety. And we're going to have some slides about this, but safety like hemolytic activity, production of the lactic acid, we're checking for the antibiotic resistance. Because when we talk about the lactic acid bacteria, always we think about they're good guys. They are beneficial, they're good, not all of them. Okay, genus Streptococcus. Which one coming to the brain first? Which one? Thermophilus, right?
Yeah.
Because of yogurt production, right? But then it's only 3 streptococci that are considered as a safe. Thermophilus macedonicus, actually the Lyticus species macedonicus, and salivarius. The rest is pathogen. Streptococcus pneumonia, need to convince you that it's a pathogen. The word is already telling, related to pneumonia. So most of them could be very dangerous, some of lactic acid bacteria, not all of them. Enterococci. Enterococci resistant to vancomycin. It's a nightmare in the medical healthcare because they are problem, enormous problem. So you see, it's not all of them are nice, but some of them are very good. But then we need to check for this. Then how we can apply them, these microorganisms? Okay, we can use them as a counterculture, obviously, all the fermented food. By the way, something interesting, India is a very rich of fermented food made by bacillus, not by lactic acid bacteria, but the genus Bacillus. And several of these products was having very good health promoting properties. And some of these bacteria producing bacteriocins as well. They're producing antibiotics, they're producing surfactants. They're really extremely good metabolitic factor because they're producing several. They've already been explored, the bacillus in the pharmaceutical and chemistry and biosynthetic processes, but they are really a huge potential to make a new probiotics because remember, most of the bacillus, they're spore forming. Lactic acid bacteria are not, but bacillus is spore forming. So you can actually produce the spores, you can keep them for the longer, and you can apply them as a probiotic much more easily. And it's one advantage, especially in the animal industry, production of farming, they're using bacillus producing probiotics for the chicken, for the ruminants. They're using even for the human application. Bacillus coagulans, that is changed the name, now is Candrexia coagulans. It's one very popular probiotic, not lactic acid bacteria, but bacillus. And it's having application in several and authorization in several countries. Even way Santos, he ran away. He got bored with me and ran away. Okay, Santos, what we got here? It's another group of microorganisms that as well can produce bacteriocin, and some of them even can be considered as a probiotic. Especially for the agricultural applications, very often bacillus is much more applicable because lactic acid bacteria are difficult. They want a rich system to grow. Bacillus is much more simple, and they can grow much more easy in the agricultural applications. So some of them of lactic acid, but well, we stick now to lactic acid bacteria, they can be considered as a probiotic or postbiotic. And we're going to discuss these two terms a little bit more details later. actually I think on the third or fourth day. Technological properties of this lactic acid bacteria in the food production, improving sensory characteristic, making products more pleasant, extending the shelf life. Why? Because they produce acid. Salmonella-like acid? No. Salmonella pH go below 4.2, 4.3, it's already not very happy. And lactic acid bacteria, especially formal lactobacillus genus, pH can drop to 3.7, 3.8. Salmonella is, well, you cannot kill the salmonella, but salmonella will not multiply. It's already something because salmonella is a big problem. Then improving the color stability. Some of the metabolites produced by the lactic acid bacteria, they can have influencing indirectly the colors of the products or even improving the texture of the food products. So let's see, how do you like yogurt? Thick or liquid here? Well, in Bulgaria as well, yogurt needs to be thick. In Brazil, they wanted liquid. I don't understand why. Reason, the lactic acid, Lactobacillus bulgaricus, responsible for fermentation of the yogurt, is not growing very well in Latin America. I don't know, must be nationalist this strain and don't want to grow there. So they actually adapted consumers to like it more liquid. It's okay, it's not bad. But again, if you use a leuconostoc that produces hexapolysaccharides as a co-culture, you actually can improve the texture of your fermented dairy products because hexapolysaccharides made it more thicker. Well, another important point, if we talk about yogurt, yogurt is only called yogurt when it's fermented by Lactobacillus delbrutis subspecies vulgaricus, and in collaboration with Streptococcus pneumophyllus. All the rest is fermented milk. So this is important point. Again, very often people going in this error when they submit the paper, they ferment it with a different, even bifid or whatever, and they call it yogurt. Sorry, no, it's fermented milk. So it's a kind of terminology. But then bioactive compounds produced by this lactic acid bacteria could be variety and variety. You can have it acid, obviously it's lactic acid bacteria, so they will produce lactic acid. They can even produce different acids, depends if they're homo or heterofermentative. They can produce hydrogen peroxide, carbon dioxide, some of them. Heterofermentative, they do carbon dioxide. And this can give this fizzy, who like a kefir? Or no one like a kefir? Kefir have these bubbles because of production of carbon dioxide. Well, most of them is because of the yeast involved in the fermentation of the kefir, but some of them, some heterofermentative lactic acid bacteria can help in this production. Low molecular weight metabolites, several of them, like reutherine, it's not a bacteriocin, it's not antibiotic, it's a reutherine. It's completely different structure, but they are there, they're produced by most of them. Actually, they say it's only Lactobacillus reuteri producing reutherine, but again, some Lactobacillus fermentum, then now it's Lactobacillus fermentum, as well can produce it, reutherine. We have the diacetyl. Diacetyl is very important because diacetyl have the kind of antimicrobial properties. Even in the beginning of 20th century, they was using for treatment of tuberculosis, but you needed a lot to treat. So actually having some side effects, but again, it's in the beginning of the 20th century, there's antibiotics. No, the people don't have choice. They look for something that can inhibit the Mycobacterium tuberculosis. Several enzymes produced by the as a bioactive metabolized by the lactic acid bacteria, of course, bacteriocin, and again, interestingly, proteinase inhibitors. Why I put them separately and not part? Well, they inhibit the growth of the microorganism. But very often, if you detect activity of bacteriocin against viruses, it's not the bacteriocin himself. It's these proteinase inhibitors, because in this way they're able to inhibit the growth of the virus. Actually, not the growth, multiplication of the virus, because the virus go, inject the DNA or RNA, whatever virus you have, and he needs to produce all these components inside the host. But since you have these proteinase inhibitors, they can inhibit some of the proteases involved in the reconstruction process of the virus. And what will happen? No virus. Well, we have some examples. We found some protease inhibitors and some bacteriocins that work against herpes virus, that work against measles, polio virus. And unfortunately, we tried against COVID, it was not working, so we don't have panacea. You cannot kill everything. But again, we need to try, we need to explore. By the way, what's the problem with polio in India? Polio virus. It's a problem because in Brazil it's starting to coming back. It's really starting to coming back. The problem is we have the vaccine, no problem. The problem, people don't want to do the vaccination. And if the polio is coming to the child, this child will be paralyzed for the rest of his life, but this will ruin the life of the family as well. It's a very social and medical problem, not just a simple problem. Well, something again very classical, very typical, but why it's important. This is the difference between gram-positive and gram-negative. But actually, this is very important when we're going to discuss mode of action of bacteriocins, because of this difference on the cell wall, bacteriocin actually from gram-positive can work against gram-positive, and bacteriocin from the gram-negative will work against gram-negative. And this is actually something that it's a big challenge for the science, because if you develop the medical formulation, you want to kill all the pathogens. And you want to use it such as the more simple one, more safer one producer, but you want to kill more dangerous one. And the problem, most of the pathogens, they're gram-negative. So how are we going to kill them? Well, don't be so disappointed, because Staphylococcus aureus, it's a gram-positive. It's a really a big problem. If you think about Staphylococcus aureus can be involved in the osteomyelite, can be involved in the skin disease, can be involved in the food poisoning, can be involved in the meningitis, can be involved in the respiratory disease. So technically, any part of our body could be attacked by Staphylococcus aureus. And the good point, bacteriocins from gram-positive, sometimes we need to select the appropriate one. They can kill gram-positive Staphylococcus aureus. But again, look the structure, look the formation of the cell wall. All these things actually have importance later when we're going to discuss the mode of action of the bacteriocins. And gram-positive, you have the peptidoglycans, quite simple structures. Well, down we have some proteins, polyphospholipids, etc. But again, on the gram-negative, it's much more complexity. But again, you have lipopolysaccharides, and this could be a good receptor. We have some proteins in the other membrane that's going to be a good receptor. And this is actually important for the mode of action of the bacteriocins. Okay, I have a question. You want me to try to do the 2-hour lectures in one block, or you want to have a break in between? How do you prefer? It's a democracy. You can say, but then I will take a decision. Can we do it in one block? Yes. Okay, no problem. So just please let me know when it's approaching time to finish, because I will continue. Tomorrow we'll continue when we stop today. We'll continue tomorrow on this stage. But again, you need to give me a sign when I need to stop for today. Okay? Deal? Okay. Some examples here with the morphology. You see, it's a kind of interesting microorganisms. Sometimes I say actually they're beautiful when you observe them in the microscope. But now it's coming some interesting points here. When we're talking about the fermentation process, it's actually transforming the carbohydrate to the energy. Fermentation is not something that microorganisms want to do for us. He wants to win the energy. And we have endogenous electron receptors in the fermentation. In the respiration process, that is completely different. You have exogenous electron receptor. So it's very simple. Normally, fermentation is an anaerobic condition when there is no oxidative phosphorylation to maintain the production of ATP. This is a rule. In anaerobic condition, there will be no oxidative phosphorylation to production of ATP. During the fermentation, pyruvate normally is metabolized to the various compounds, and in whole lactic fermentation, production is only going to pyruvate and then to lactic acid. It's rule. In a heterofermented fermentation, you can have the different final products and different acids, and even alcohol that could be produced. Well, in the yeast, it's a little bit a different story. But again, in the lactic acid bacteria, homofermentative process, it's going down to the lactic acid. In heterofermentative fermentation, you can have acids of different variety, but lactic acid will be for sure. There's no way to escape. So we have these pathways, very simple, already had this in the books of microbiology. The professor asked you to remember all these pathways. I don't want to remember this because it's more important. I mean, okay, you have glucose 6-phosphate, fructose 1,6-biphosphate, but you have glucose. You're going to glyceraldehyde 3-phosphate, then going down to pyruvate, going down to lactic acid. Glucose, pyruvate, lactic acid. In heterofermentative, you can have the glucose, but you can have pentose as well. And very important, are you going to be university professors or you want to go to the industry? Who wants to go to the industry? Hence. Industry give them money. They pay well. Who wants to be a professor in the academic? Well, you need some time to show the students heterofermentative in the lab. Normally take one leuconostoc. For sure, leuconostoc will produce this gas, CO2. You even take the glass tube, you put on the vortex, you have a huge amount of foam. It's already showing the presence of CO2. If you have production of CO2, obviously you have the heterofermentative microorganism. And this will help you later in the taxonomy. Normally people always complaining that it's too costly, too much work, this biochemical, physiological tests, but we need them. Because very often when the people talk about the identification of lactic acid bacteria, everyone, okay, genetic. Yeah, genetic. Try to do the identification of enterococci by genetic methods only, not going to work. 16S sequencing is a very good tool. It's helped a lot. But the problem, when you do the sequencing of 16S in enterococcus, try to blast the results. What you're going to have? 99.9% enterococcus fecalis. 99.9% Enterococcus munti, 99.9% Enterococcus caselinoflavus. Which one? The problem, the 16S segment for the enterococcus is very conservative. It's not helping. You need to go back to biochemistry. You need to go back to the physiological tests in order to have appropriate identification of enterococcus. Same thing with not lactic acid bacteria, with bacillus. And you're going to be scared. Soy milk is popular in India or not? Okay, soy milk very often is contaminated with bacillus. The fact because it's a plant material, plants go where? On the ground, no way, and the bacillus is very present in the ground. So you have contamination with bacillus, and when you bacillus make spores, so you pasteurize the soy milk, you put in the Tetra Pak, you put on the market, and 3 months later you see the Tetra Packs look like a balloon. Well, bacillus grow, the spores survive the pasteurization, they grow. You do the test in the lab, you came like scary, Bacillus anthracis. It's not, it's Bacillus subtilis most of the time. Well, could be Bacillus cereals, that is not a good one, but very often it's Bacillus subtilis. Problem is the same thing. 16 nets in Bacillus giving the very same results for several Bacillus because it's a very conservative. So obviously, again, genetics is a good tool, 16S is a good tool, but it's not it's not a cherry of the cake. We need to know basic biochemistry, we need to know the physiology of this microorganism because it's a complex way to do the appropriate identification. And then, very often people making this error, they say the lactic acid bacteria grass, generally recognized as safe. Well, more or less, but general, just a general, not a real. The problem is such a big group of microorganisms, lactic acid bacteria. As I mentioned to you, most of the streptococcus, they are lactic acid bacteria, but they are pathogens. And they are very dangerous pathogens. Half of enterococci, they are carrying vancomycin or methicillin resistance, and they are a problem. You don't want to go in the hospital and to catch vancomycin-resistant enterococcus. No way, because what antibiotic we're going to use now with you? Because vancomycin was considered like 10 years ago was a last option. Now we're having some new, but again, it's not easy. Then you'll be surprised, some of the species that are considered traditionally safe, like the Plantabacillus plantarum. It's considered traditionally as a safe microorganism. Some of them associated with the inflammation of the heart, human heart. Some of them can carry virulence genes. I have some examples later doing a kind of screening for virulence genes in the different lactic acid bacteria. And you can really surprise to see how much virulence genes could be present and can make this strain very nasty and good candidate to receive the name of Falimadin-law. Well, another point that needs to be always taken into consideration, production of biogenic amines. Biogenic amines, it's, we're going to have more about this, but now very shortly, they are decarboxylation of the amino acids, normal physiological biochemical process in the transformation of the organic substances. The problem, some of these components actually could be toxic, and they can be influencing even the brain function, some of the biogenic amines. So if you have, let's see, non-fermented food products, and there was a biogenic amines production there, well, there may be a little bit, but the problem is when you have fermented food products. These fermented food products, especially probiotics, they need to be alive. They need to have a live microorganism. And they can produce all the time biogenic amines. So they can accumulate in the product. And in fact, in the time you can have a product that is actually coming to the toxic levels, these biogenic amines. So this is another important point when you do the work with the lactic acid bacteria. We need to take care to don't select none of the microorganisms that could be potential problems. Well, if I develop new probiotic, I want to put something on the market that is safe because I want to have sleeping in the night conscious. I don't want to have nightmare that someone gets sick of my product. So, right. Let's be honest, and money is not top thing in the life. First of all, we need to be responsible, and we need to produce the things that will give the benefits, not negative points to the consumers. Now, lactic acid bacteria I already mentioned, but let's repeat again. They producing organic acid, diacetyl, hydrogen peroxide, carbon dioxide, bacteriocin, low molecular weight, and they was used these antimicrobial products produced by the lactic acid bacteria since the human civilization exists. All the fermented food products produced all around the world in all the civilization, they involved lactic acid bacteria, they produced these antimicrobials, they protected the product, they've been used by the traditional medicine and even with the modern medicine because these days probiotics with the bacteriocynogenic property and producing other antimicrobials, they are actually having more and more interest from the industry because they can have the better application. But coming to the question, what is the bacteria? Since I was using this term several times, and still now I was telling nothing about this. Well, definition that is accepted is like almost 30 years, more now. It's saying this is the ribosomally reduced polypeptide. Well, why ribosomally important in this definition? Because you can have it antibiotics with a protein structure, but they are produced and then they have the enzymatic modification in the cytoplasm. In the case of the bacteriocins, they are produced in that way, and they work as a produced. They will have very slight modification after that, only with one specific group. And normally, they are active against closely related microorganisms. Meaning, again, remember I was mentioning in the beginning, bacteriocins from gram-negative, they kill gram-negative. Bacteriocins from gram-positive, lactic acid bacteria, they will kill pathogens on spoilage of gram-positive. Well, this definition actually needs to change, and we're already suggesting some changes, because if you look the history of development of the bacteriocin, they start in 1925. 1 century. Bacteriocin already on the scientific literature. First one actually was collicins, produced by Escherichia coli in 95. Then a little bit later we have bacteriocins from gram-positive. And then originally they was introduced as a biopreservation tools. Well, very good, so we can protect our food products. Well, our grandmother was protecting them because of fermentation and production, but now we know what is going there, why they work this fermentation process, why they're protecting the food product. Well, later it was discovered that some of the bacteriocin from lactic acid bacteria, gram-positive, in fact can kill gram-negative. Well, what's going there? Why it's working? Why it's happening? They realize, in fact, very often it's not a simple interaction between bacteriocin and gram-negative, but there is a synergistic interaction between bacteriocins and some surfactants. Well, who work with the lactic acid bacteria again here? Using MRS media? Yeah, they work very well. What is in the MRS? Twin 80 surfactant. And twin 80 together with the bacteriocin actually working together in the synergistic effect. In this way, first twin part of the media actually will make a change in the surface of the gram-negative, and then the bacteriocin coming and do his job. In fact, it's not just a simple bacteriocin, but then we get a little bit further. We wanted to purify the bacteriocin because we want to go everything out of the surfactants. Well, we know if you want to kill in the pharmaceutical products, you can mix not only bacteriocin, you can put the surfactants, you can put everything that you want, you make a complex, because the pharmaceutical product is not a simple single molecule. It's a complex preparation that everything needs to interact and do the best way of the job. Stability, efficacy, etc. Well, we purified the bacteriocins because we had one produced by Enterococcus multi that was very good activity against gram-negative. So we purified, and we had a single peak on the HPLC. We do a mass spectrometry, confirming we have the pure product. Still work on the gram-negative. Well, the problem is we stopped because the bacteriocin been taken as a patent from the medical pharmaceutical company, and there was that we're not allowed to publish any more results in mechanism of action, but they wanted to know what's going on. Well, it's very simple. We discovered that we are attacking different receptors in the gram-negative, and attacking this receptor, bacteriocin having the option to do his job even against this gram-negative. But if the gram-negative don't have this specific receptor, they cannot work.
So the patent was on the sequence of the
bacteriocin and application. Well, we're not allowed. I mean, I don't know. Some countries they allow, but in most of the world, we are not allowed to patent microorganisms because something that is from nature. We cannot patent something that has always been there. But we can patent application. We can patent the products produced by this microorganism. The patent also peptide and the killing this gram-negative pathogens. By the way, companies from India.
That's the US patent?
No, international. Because the work was done in South Africa, together with this Indian company that had a branch in Australia, in South Africa. I think I'm not allowed because of the record to say the name because we'll be like advertising the company. But it's a company from here. If they pay me the royalties almost 20 years, it means they're selling the product. But they was very strict to don't release the mechanism of action. It's I'm releasing. It's a different receptor for the bacteriocin that it's managed to attach to this receptor, but I'm not giving structure of receptor. It's helping something. Well, but I don't see the problem with this. Well, anyway, later some of the effect of the bacteriocins was shown to work even against Mycobacterium. Now, okay, I will do something. This is matter. Okay, let's think about something. Now we're in the contact like 1.5 meters. If I'm having tuberculosis, she's already in trouble. 1.5, 2 meters, it's enough social distance to transmit. Hello. Congratulations, you have it. Okay, no, don't worry. I need to do a test every year because of the safety issues and work in the lab for the TB. But the TB, it's a big problem because you can catch it in the social contact. I'm not sure what's the level in India of TB, but when I was working in South Korea, they was having a very strict rules for only not Europeans. They was not checking for TB compulsory. Rest of the world, they was checking. In Zimbabwe, more than half of the population is sick, not caring, having a medical problems. And imagine if you have the bacteria that can reduce, not kill, just reduce the Mycobacterium tuberculosis. You already have something that helps. Again, we don't want to say that the bacteriocin will save the world. This is not a new antibiotic. It's not a completely things that will replace the antibiotic, but they can help to reduce. And another question, Santosh told me that it's about 50% subscribe for this course. Okay, let's do the calculation easily, 60. 1/3, one of every 3 persons actually carrying Mycobacterium tuberculosis in the body. This bacteria maybe will never develop, never. Will stay sleeping in your body for the rest of your life, but it's kind of high. It means in this place about 20 people, most probably having a Mycobacterium tuberculosis inside the body. Okay, don't be panicked. This bacteria will sleep there for the rest of your life, but it's a kind of common. And if you can have it again, probiotic or with bacteriogenic property that can inhibit Mycobacterium tuberculosis, so even Mycobacterium bovis or Mycobacterium avis, because they can be transmitted to the humans, it's already some positive things. Inhibition against some fungus. We're having this work showing that some of the bacteriocins can work against fungus. You see, already putting away of this whole definition, that's only closely related. You can inhibit some more than just a closely related. You just need to have a propertive model. You need to have very often using strains that are not pathogenic as a model in the lab, because we need to respect the safety in the lab as well. And then you can explore this specific interaction and actually develop new pharmaceutical products for the application in the human veterinary or agricultural applications. Synergistic interaction. Bacteriocins work together with antibiotics. They can help each other, and in this way you can reduce the level of the use of the antibiotics. Anti-candida effect as well was reported. And Candida albicans could be a very serious vaginal infection. And treatment, in fact, with some bacteriocins and probiotics producing these bacteriocins could be a good option. Antivirus activity, I already mentioned, polio, herpes, measles virus can be treated. We have 99% reduction of herpes virus, 50% reduction of polio virus. It's not a medicine. Again, please don't ignore the vaccine, especially when you have the vaccine, but this additional treatment never, it's need to be ignored. Even control of cancer cells. There was a very good review published by 2 scientists from India, Kaur and Kaur. Must be wife and husband or brother and brother, I don't know. But they was really pointing this on this interesting effect of some bacteriocins. And interestingly, they can attack only the cancer cell, but they don't attack the healthy cells. We was working in Brazil on some project, and we was needed to check the cytotoxicity of some of our bacteriocins. And we used the cancer cells because it's much more easy to work with them in the lab, right? Who work with the cells? No one?
Cell lines, we
we
don't have. Few labs, they have cell lines.
Yeah, but it's, I mean, when we work with the cell lines, it's much more easy. The cancer cell lines, they grow easily, they develop easily, they don't want to die. Much more easy. Healthy cells, it's more difficult. But interesting, we have a very high cytotoxicity against cancers of breast cancer, cells of breast cancer. And it was like, okay, they are cytotoxic. But then colleague of mine have it, okay, let's try with the healthy cells. No cytotoxicity. And then we repeat it again and again. Very high activity against cancer cells of breast cancer, and almost no cytotoxicity against healthy cells. And then starting digging the literature, then was a report here, another one, then come this review from this scientist from India. There was summarizing all these things, and now it's getting a trend in the research because some of the bacteriocins have the specificity of the interaction with the cell of cancer, because the cancer cell, they have a different charge on the surface, they have some modification in their surface, and the bacteriocins can easily recognize them and actually kill them. And now even signaling molecule and quantum sensing, they involved in all these processes. So the timeline, more or less, again, 95 polycines, first from the lactic acid bacteria, 28, 33 in niacin was discovered, when authorized in almost around every country in the world. Received his name in 47. Why is such a big difference in the time? Why needed 40 years to receive the name? What's your opinion? Why? What happened in this period? Second World War. Do you think people were thinking about the niacin? They will look how to survive at that time. So this was a reason to take a little bit longer. Well, authorized in the Europe, in England, in the World Health Organization, US, etc. And then coming for this variety of applications again. Antimicrobial produced by lactic acid bacteria can have antimycobacterium. It's a good thing to explore. Antiviral properties then could be bacteriocin, could be protease inhibitors. Antifungal properties very often associated with different acids, especially phenyl lactic acid could be very often responsible for this. Antibacterial classes, different classes, bacteriocins and different other molecules. But important thing, if you look here, when you have the small amount of the bacteriocins, normally small amount is responsible for interspecies communication. This could be quorum sensing, could be quorum caching, or even between interspecies communication, when it's a small amount. Look like in the nature, bacteriocins being developed by the... Okay, sometimes I'm using this example. People that are very religious, okay, the God created bacteriocin. People that are very anti-religious, the nature created evolutionary process. How do you want, you can do it in your way. But important thing, when normally they're being created to use this interbacterial communication, they was not created to kill other microorganisms. That was much more important in this communication area between different microorganisms. Lately, when they are having in the highest level of the production, they can have bacteriostatic, bactericidal, or bacteriolytic effect. And when it's a little bit higher concentration, then you can have anti-cancer effect, they can have eukaryotic viruses, inhibition, or even effect against viability of spermatozoids. So it's getting more and more complex, but again, in the beginning we was exploring these antibacterial properties, but with the development of science we went to the communication and as well not typical killing properties of the bacteriocins. And develop of the history of the bacteriocins. This is taken from Scopus, and from, let's say, how much this will be, 55, 80, last 80 years. When you put the term bacteriocin or bacteriocins, because very often in Scopus you need to put singular and plural to have better results, you see how exponentially growing. Well, it's here, it's even more, because the data was been taken based up to the 24. But again, if you cross the word bacteriocin and antibiotics, or bacteriocin and cancer, here bacteriocin, antibiotic, bacteriocin and cancer, you see as well develop of the research projects, publication of papers taking care for this. Well, here it's not too much, it's like 60-something papers per year, but it's already starting, because it's showing the potential of these products against cancer cells. How much time I have? What time I need to finish?
We have lunchtime and another 15 minutes.
Okay, at 1 o'clock I need to finish. Okay, so we can talk about mode of action and tomorrow we continue. By the way, in the afternoon, Santosh promised you to try to isolate your own probiotic. It's exciting because you will find that in 1 week you will have something in your hands that giving a potential. But then you need to study on this, and we spent like 2-3 years to show what you have exactly. Okay, how they work. Most of the bacteriocins, actually, they're using associated with the proton motif force. And they can make a pores inside the cell wall. And in this way, they can actually interact with the protein motif force. This is actually potential energy generated by the electrochemical gradient established between the sides of the membrane. But if you make a pore inside the membrane, obviously you cannot anymore control this balance. And so in some cases, this will interact with anchoric molecules, and they facilitate the interaction of antimicrobial peptides in general, bacteria more specific, with the plasma membrane. This actually increasing problem with stability, and you have it evolve lipid 2 and system mannose phosphotransferase system. So lipid 2 and mannose phosphotransferase system, they actually are very important because they can serve as a receptor for some of the bacteriocins, and different transporters as well can be involved in this process. It's interesting that some of, okay, do you know how works the venom of the cobra? Do you know how it works? They block some channels and they interrupt the transferring of the signal, they interrupting with the stability of the cells. Same thing. If you use the bacteriocin, they can interact with these channels, specific channels on the cell surface, and in this way they can actually interfere with the cells on their integrity. In the laboratory, sometimes using the poisons from the scorpions, from the different snakes, including this one that is like naja.
That's a cobra?
Yeah. Using the poisons for them, because in this way we modulate the different channels in the cells, and we can see if the bacteria is attacking the same channel or not. And we can actually make a conclusion how the bacteria is actually killing the pathogens. I don't want to see the snake. Sorry, I had the phobia. If I see the snake here, you see me in the airport in the 10 minutes. I will run to the Delhi International in 10 minutes. Will be the new Olympic champion from this university. But important thing is formation of this interaction. And very important role is here of the lipid 2, because remember from the antibiotic courses, most of the several antibiotics, vancomycin, etc., they interact with the lipid 2 in order to make a contact and to disturb the cell membrane. And lipid 2 is actually in the phospholipid layer, and translocation of the periplastic space to the membrane of murin synthesis, okay, it's a classic microbiology of importance of this lipid. But again, bacteriocin can interfere with the lipid 2. And since the lipid 2 is important for the cell, you cannot take it away. If the cell losing lipid 2, we'll have the problem with the murin synthesis. So what's happened? Bacteriocin cannot develop resistance because the receptor is there. You cannot take away the receptor. If the bacteria losing the receptor lipid 2, she will die. But then bacteriocin is coming and recognize the lipid 2 and kill. Well, for now, we don't have such a strong resistance to bacteriocin. For now. But remember when Fleming developed the penicillin, well, everyone was happy. We finished with the disease, no any more disease. Well, 10 years later, problem with antibiotic resistance already started. Same thing, for now, we are happy. Sometimes I want to say, the development of new antibiotic, development of new medicine is a kind of a battle. We win the battle, but we don't win the war. War is still with the pathogens, is in the front of us, and will be, because obviously microorganisms are not stupid. They will develop some kind of resistance. They don't want to be just stand there, okay, kill me. I know you don't like me, kill me. No, they will try, they will find a way to protect themselves. And maybe in 50 years we will have the resistance to bacteriocins, but at least now they work, and we need to explore this. So Maureen side chain and cross links. Maureen, the final product of the lipid 2, is interacted by the complex network of amino acids chain, so everything's okay. And this is the point that the bacteriocin come and can interact. So they can be bacteriolysins, or they can be bacteriostatic. Well, this is a different story, because sometimes we don't kill them, but we push them down, and then it's coming the different mechanism of the host that can influence. It's not always that we can have the bacteria and they will solve all the problems. We need to have cooperation with the other systems of the organisms and the way to protect from the pathogens. Well, some of the bacteriocins like enterolysine A, they can interfere, they can go inside the cell wall destruction because they can incorporate in the phospholipids. But if you destroy the phospholipids, you're actually making the cells more easily to be targeted by antibiotics, they more easily to be targeted, but even by the acids. So again, not a single molecule. It's a complex of different molecules. In fact, it's the way to work better. Mannose phosphorotransferase system, PTS, is associated with the spontaneous zinc complexes. And when you interfere with this system, you block the system or you open it completely. You interfere with the metabolism of the cell, and it's work. Then you can have the bacteria, especially if you're gram-negative, they can interfere with the RNA polymerase. They make a simple complex with the RNA polymerase, but once this protein, bacteriocin, make a complex with another protein, with the polymerase enzyme, well, polymerase is not working anymore. You block the RNA polymerase, what will happen? Not going to nothing good. Or DNA GRAS, again, one enzyme combined with another protein, two proteins making a complex. Complex, it's interrupting. It's like an enzyme inhibitor. They work in a very simple way.
So what is the concentration at which they work?
Well, this is an interesting point. They work very well even in nanomolar, very small amount.
So when you were testing it against the cancer lines, what concentration was that?
Well, the question is, when we work with the bacteriocin, we don't checking their concentration as a milligram protein or nanogram, whatever. We're checking them as arbitrary units, like enzymes.
So was it a pure preparation?
So normally we're using our own preparations, normally purify them. Very often as a semi-purified, it's much more easier and cheaper. And you don't need the equipment. You can do it with a very small column and syringe in the lab. Only you need to put 5 students because you need to do drop by drop, drop by drop. But it's cheaper than by the H+C. Well, in one moment we need to go to the pure material. But normally, even in the 50 arbitrary units per ml, they're having a good effect. Very often this is corresponding to some microgram of protein, enough. Bacteriocins can be combined with antibiotics again, because they work in a similar way, but they're having a different targets. And if you target pathogens on one side, in one receptor, you disturb the membrane, and then coming antibiotics, they're doing a completely different mode of action, obviously will work even better. So vancomycin, niacin, and nisin, they work together. There's several work in the treatment of methicillin-resistant Staphylococcus aureus. We're having some microcins and chloramphenicol. There was as well some gram-negative bacteria can be worked. And in general, when we talk about their mode of action, we need to keep in mind the 2 principal ways. First way, the bacteriocin is coming, there is a lipid 2, and terminal of the bacteriocin will recognize the lipid 2 because they can make a recognition, and the C terminal will go inside. And it's interesting that most of the bacteriocins that have an anti-listeria effect, they have a very constant N-terminal part of the molecule. And they will always recognize lipid 2. This is why listeria is very easily to kill by some bacteriocins. Some of other, like class 2 cytosines, they go in and by electrochemical interaction, they will interfere, they will incorporate as a protein inside the membrane. Remember the membrane of gram-positive, gram-negative, they have proteins in there. And they have this kind of mimicking, like, I'm your protein, I'm protein from your membrane, and they can come and incorporate. Or another one, that is one that one mentioned that interfere with the phospholipids, bacteriolysins, they go, they make this complex with the phospholipids, and they destroy completely the cell wall. Again, too much work is done, but we're still on the top of this iceberg. It's a lot to explore, especially in the mechanism of interaction, because, as I mentioned, sometimes a specific receptor in gram-negative could be responsible to have this contact with the bacteriocin, and bacteriocin from lactic acid bacteria can interact, and they recognize the receptor, they go in the membrane, and they destroy it. In Sao Paulo, if you leave your house with the open door, you find nothing. It's quite sad, but crime existing. It's not too bad, but, well, exists. And the same thing, if you open the bacteria, you open the pore, you will lose everything, for sure. And different method, different receptor, interaction with RNA, with proteins, enzymatic proteins associated with the cell life of the cells could be a very good option to see how they work. And then, finishing really quickly in a few minutes. So now the bacteriocin is produced. And normally bacteriocin is produced, this is the bacteriocin, this is the leader peptide. It's a complex molecule, but in this way the molecule is not killing nothing, because it's two parts that are together. In this form it's not active. But when it's going to be the ABC transporter, one part it's losing, and you have active bacteriocin, after that they will go on. But then, interestingly, these bacteriocins can actually interfere and can activate production of the immunity protein, so that bacteria can protect themselves from the bacteriocin, because bacteria maybe have lipid 2 as well, and if has lipid 2, this bacteriocin maybe will try to kill his own producer. Well, it's not good. So, but then you have the activation of the immunity protein because of this interaction. Then some of the bacteriocins can go to the neighboring cells and recognize this histidine protein kinase. And recognizing will activating this death for losing the phosphor from ATP, DTP, dephosphorylation. Well, losing phosphor from ATP to DTP, and this phosphor, in fact, will be serving as a responsible regulator for activation of the gene on the DNA level to start producing activating all the operon responsible for the production of the bacteriocins. So one bacteria producing the bacteriocin go to another bacteria from the same, his brother, sister, whatever how we want to call it, and by this system will activate bacteriocin production in the next one. Another interesting point, some of these inductors actually coming from the pathogen. Listeria have the surface proteins that are able to be recognized as a pseudobacteriocin by the cells and will start inducing production of the bacteriocin. So, okay, look guys, we're having a listeria around. We need to produce our bacteriocin to protect against listeria. And this circle is actually going and going and going. But then some of them, bacteriocin, they have this leader peptide. This is the bacteriocin leader peptide. And when it's going via the ABC transporter, they need to cut and liberate the real bacteriocins. But some of them, they are leaderless. They don't have leader peptide. They are produced, but then this interesting point, on the N-terminal, they always have methylated methionine. And this fact that have this methylated methionine protect the old producer by effect of this on bacteria. Let me just quickly. Yeah, I can finish in 5 minutes. Okay. Now you have it this schematic way, so you can at the top you cannot see it, but this is the same bacteriocin there, and they have this leader peptide in the front. They need to be liberated. After that, the bacteriocin very often can make these disulfide bridges between some non-specific amino acids. You can have metal lanthanoin and beta-methyl-lanthanoin, not proteic amino acids. But you can make these disulfide bridges that will give a specific structure. But then you have this cleavage that will be done here. Leader peptide will be liberated when going via the ADC transporter. And when it's going out, you're going to have only this part. In case of the lysine, lactocin, another lactocin, but going out already with these disulfide bridges. But this structure actually is important because only in this case, the molecular of the bacteriocin can have the specific configuration to recognize the receptor. If it's not this structure, no recognition, no effect. Then it's by the way, I'm going to provide you these slides. Some you can give it to them, right? You need to decide how we're going to distribute the slides. You have in here, it's like a story. Again, you have interleucin the various genes associated, various product of the gene expression. But anyway, it's important to see that you have a activation of the specific protein. Its fact is coming to interleucin F, that is induction factor. This induction factor needs to make contact with interleucin K, this kinase enzyme. They will activate interleucin R, that is a regulation factor. And only in this case, expression of the entire operon will be available. So no choice, you need all these steps in order to produce and bond. Classification of the bacteriocins could be very different. In the past, we have several classifications. Very often people just put a new classification to have more citation. Well, it's okay. But most of them, they say antibiotic class 1, it's a classical one. They are smaller than 5 kilodalton. They have lanthanoin and beta-methyl-lanthanoin and some unusual amino acids. They need to make disulfide bridges in order to have the active molecules working. There are several of them. They are cationic normally, negative charge. Some of them could be positive charge, but majority is negative. Some of them could be even globular. Then you have class 2, the smaller than 10 kilodalton. So there is a nightmare sometime in the protein work because small protein is more difficult to work from some point. But the good point, they're thermostable because they're small, they're nothing to lose in the high temperature. Very important part is 2A pediosin-like bacteriocin. They have this activity against listeria, and they have always this conservative amino acid. So you can easily target this part of the molecule on the genetic, on the protein level. You can easily discover if they have this fragment on the N-terminal. There is some bacteriocin, they're a nightmare for the work because there are 2 components, 2 proteins, and they need to be together to work. If you separate, they don't work. And when you purify them, how you can reconstruct them? It's difficult, but it's fun to work with them. Again, this SEC pathway, this one, they don't have this leader peptide as well. And worst scenario, if you work with a bacteriocin, you have the bacteriocin that is a complex of protein and non-protein molecule. Because you need to have a protein on one side, you need to have carbohydrate and lipid to get together in order to work. You do the protein purification, you lose the protein, lipid. You do the protein purification, you lose the carbohydrate, and then it's not working. So how you know that you purify? But again, it's more fun to work. Then it's a classification from Paul Potter. He just said, very simple, class 1 antibiotics, class 2 non-antibiotic, very simple, but it's a kind of difficult. And I like much more this of John Tack from New Zealand, because it's again, antibiotics, they can be linear, they can be globular. Niacin is coming here in this group. Class 2 small non-modified peptide, because they have this small modification, this disulfide bridges that will appear and non-specific amino acids. And here class 2, they are non-modification, pediaxylite. You have class 4 that is cyclic peptides because none of them are linear, some of them could be cyclic. And you have class 3, very big proteins that have this antimicrobial activity. Okay, let's stop here for this morning. And tomorrow we continue, actually not tomorrow, afternoon, we continue with some practical. And when we do the practical, actually you can see why all this information is actually linked to the manipulation. Questions can be after lunch?
Yes.
Okay.
Thank you very much, Mr. Ravi. This was very like this afternoon session, we will be having more interactive sessions. Welcome the Department of Genetics seminar. He will interact with each of the participants, their interest, how to about hands-on training, how you can isolate these probiotics, how you control it. So we will have more there. Go for have a quick break after the lunch with the Department of Genetics. Thank you very much for your attention.